Okay, so now we're going to start on chapter 3, which covers the cellular level of organization. So this is all about the structure and function of cells. This is a very long chapter. I'm going to try to go a little faster than I usually do because I don't want this video to get too long, but this is probably going to be one of the longest ones of the semester. So we'll start off with an introduction to cells. Cytology is the study of cellular structure and function. We mentioned this in chapter 1. It is a division of microscopic anatomy. The cell theory states that cells are the building blocks of life, so all life is made up of one or more cells. All cells come from the division of pre-existing cells, so cells cannot just magically appear from nothing. Cells are the smallest units of life. And cells maintain homeostasis. And so later when we talk about tissues, organs, organ systems, and the entire organism, remember that all of that reflects the combined coordinated activity of many cells that are working together. So the basis of all of the functions of the human body come down to the cells, and that is why we have a chapter on the cells, and it's actually why it's a pretty long chapter. So there are two broad types of cells in the human body. There are sex cells, also called germ cells or gametes. These are the sperm cells in males, or in the oocytes or eggs in females. Sex cells are unique in that they only have half of the genetic material as the other cells in the body, and that is so that one can come from the man, one can come from the woman, and when they unite, then they create a new organism with the full complement of DNA. Somatic cells are all the rest of the cells except for the sex cells. So somatic means body. So these are your body cells. And this chapter focuses on a typical somatic cell. So here is an image from your textbook. So we're going to go through a lot of the structures that are shown in this image. Again, that's why this is a somewhat long chapter. So this is just an overview image and an image you have in your textbook to get familiar with. We're going to talk about many of these different parts and some terminology to go ahead and get in your brain for a lot of the rest of the semester, actually. Extracellular, when you see the word extracellular, we're talking about something outside of the cell. So extracellular fluid would be fluid outside of the cell. And intracellular would be the inside of the cell. So anytime you see a reference to an intracellular, something like intracellular fluid, that is inside the cell. In this section, we will talk about the plasma membrane. So the plasma membrane is the outer membrane that surrounds all cells, and it functions to physically isolate the inside of the cell from the outside of the cell. So just like we have skin that separates our insides from the outside environment, the plasma membrane works the same way for cells. It also allows cells to regulate its exchange with the environment, so it allows the cell to control what it lets out and what it lets in. It also provides sensitivity to the environment, so there are receptors on the surface of the cell that lets the cell sense the conditions around it. And it provides structural support, so it provides a stable structure to surround the cell and hold the cell contents on the inside. The plasma membrane is about 42% by weight made up of lipids, which are fatty molecules. It's a little over half protein, 55%, and then there is a very small amount of carbohydrates in the membrane, and even though it's a very small percent, they do play a big role. So let's talk about the membrane lipids. So the membrane itself, the backbone, the basic structure, is made up of phospholipids. Phospholipids are little molecules that look similar to this little picture up here. They have a hydrophilic head and two hydrophobic tails. Remember that hydrophilic means water-loving and hydrophobic means water-hating. So because of their structure, they form a bilayer, which means two layers in a row, with the heads facing outside and inside where there's a watery aqueous environment, and the tails all go on the inside to form a hydrophobic middle. So you can think of it like a sandwich with an oily, lipidy hydrophobic middle, and an aqueous outside. In between these phospholipids, you can also find little molecules of cholesterol. So in this picture, they are shown by the little squiggly yellow molecules that are packed in between the phospholipids. Cholesterol provides both stability and fluidity to the membrane. So it keeps it from becoming too runny, but also keeps it from becoming too stiff. So it keeps it in the Goldilocks zone of just having the right amount of ability to move around without being too loose. So membrane proteins are proteins that are associated with the membrane. There are two classes. There are integral proteins, which are proteins that are embedded into the membrane. These can sometimes also be called transmembrane proteins because they span the entirety from one side to the other. And then you have peripheral proteins, which are associated to the membrane but not embedded in it. And remember, proteins are the workhorses of the cell. We mentioned that back in chapter 2. So proteins in the membrane have a lot of jobs. So there are anchoring proteins that can anchor to the cytoskeleton inside the cell. They can also anchor to other cells or extracellular fibers to allow the cell to position itself or move around. There are recognition proteins, and these are usually glycoproteins, which have little sugar molecules on them, shown in this image by the little purple hexagons. And these act as sort of like IDs. We call them recognition proteins. And so they allow the cells to represent each other. Enzymes are proteins that work to catalyze different reactions, different enzymatic reactions. And so most of the enzymes are usually inside the cell in the cytosol, but there are some that are embedded in the membrane itself. Then there are receptor proteins. These are proteins that have a specific molecule bind to them and cause something to happen in the cell. And we say that they are specific to the ligand that binds to them. Carrier proteins allow for molecules to be transported from one side of the plasma membrane to the other. And again, they are solute specific, so the type of molecule that they transport from one side to the other is very specific for that particular carrier. So for example, one carrier might be able to carry sodium but nothing else, and another carrier might only be able to carry calcium. There are also channel proteins, and we're going to talk a lot about these when we get to the muscular and nervous systems. Channel proteins are also solute specific. So for example, a sodium channel will only allow sodium to pass through it. And channels can also be gated, and we'll talk a lot about those later too. That means they can open and close based on different circumstances. So the membrane itself is a very dynamic structure. It is always shifting around. Your textbook says to imagine like bowls floating around in a punch bowl. And I'm going to show you a video as part of the in-class activity to help you visualize this dynamic fluidity structure of the plasma membrane. So finally, the membrane carbohydrates. These are usually associated with other molecule types, which means the carbohydrate usually has to be stuck onto a protein or a lipid. So if the carbohydrate is stuck onto a protein, it's usually called a proteoglycan or a glycoprotein. And if it's stuck onto one of the phospholipids, it's called a glycolipid. And again, in the picture from your textbook, the little purple hexagons sticking off the outside of the cell represent all of the carbohydrates. And together, all of these make up what is called the glycocalyx. So the glycocalyx is the outer layer of protruding carbohydrates. And here is another image. The carbohydrates or sugars are represented by the little blue hexagons in this picture. You can see the lipid bilayer in gray. You can see some transmembrane proteins, some integral proteins here. There's a peripheral protein. And then you can see that all of the sugar molecules together on the outside of the cell make up the glycocalyx, sometimes also called the cell coat or the coating of the cell. The glycocalyx functions to lubricate and protect the cell. It also can help with the anchoring of the cell to outside structures, which can help the cell with locomotion and moving around. It allows for specificity and binding, so the cell only binds to certain other cells that have the correct carbohydrates on the outside. And this goes back to the idea of these acting as ID tags for recognition. So for example, these carbohydrates actually play a big role in your immune system being able to tell which cells belong to you and which cells are foreign cells. So it's sort of a way of one cell being able to recognize the other as, oh, hey, you're also part of Dr. Sawyer's body, or oh, no, you're a foreign invader. So now we will talk about the cytoplasm, and then we will introduce organelles before getting into the specifics of them. So the cytoplasm is a definition for all of the material that is located between the plasma membrane on the outside of the cell and the nucleus of the cell. Cytosol is the term for the actual intracellular fluid, which is the fluid inside the cell. And then organelles are structures that perform specific cellular functions. Organelle actually means little organs. And so you can think of organelles working for cells the way our organs work for us. And so the cytosol and organelles together make up the cytoplasm. So this is something very important. Put a star next to this because this is going to really come back later when we talk about how nerve cells and muscle cells work. So cytoplasm has a very different composition than the extracellular fluid. So we have an example of a cell here in this pink circle. Extracellular fluid would be the fluid on the outside. So here are some very key differences, and again, please make a note of this. Potassium levels inside the cell are always higher than outside the cell. And this wouldn't, by the way, this wouldn't be possible without the plasma membrane. The plasma membrane separating the cell from the outside is what allows for these differences. Also note that the inside of the cell has lower sodium concentrations than the outside of the cell. So the cell is higher in potassium but lower in sodium than the outside. There is also a much higher protein content inside the cytosol, and that is because there are many enzymes inside a cell doing a lot of different functions that allow your cells to keep trucking. And then you have a lot of reserves inside the cytosol, so you can have carbohydrates in there stored up for energy, amino acids floating around that's used to make more proteins, and lipids in there as well to provide energy. Both the inside and the outside of the cell can contain inclusions, and inclusions are insoluble materials. So they are like little clumps of different types of compounds. One example of an inclusion that's actually found inside cells is the pigment called melanin that gives your skin a brown color. That is an insoluble material that is found inside of your skin cells. So the organelles are the cellular structures that perform specific jobs within the cell. There are two broad classes of organelles, those that have membranes and those that do not. The non-membranous organelles are the cytoskeleton, the microvilli, the centrioles, the cilia, the ribosomes, and the proteasomes. And the membranous organelles are the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, mitochondria, and the nucleus. And membrane flow is the concept that a lot of membranes are shared among the different structures. So for example, the plasma membrane, the endoplasmic reticulum, the Golgi apparatus, and lysosomes and the nucleus all share interconnected membranes. So the phospholipids and the proteins in those membranes can flow and move from one structure to the next. So it's a very dynamic structure. In this section, we will go over the non-membranous organelles, which include the cytoskeleton, the microvilli, the centrioles, the cilia, the ribosomes, and the proteasomes. So let's first talk about the cytoskeleton. So the cytoskeleton is actually a very dynamic structure. So we think about our skeleton as something that is stable and stays relatively constant throughout our lives. But inside a cell, their skeleton is constantly assembling and disassembling. And I will show this to you in a video as part of the in-class activity. There are 4 structural types, and you should be able to differentiate between these. There are microfilaments, intermediate filaments, microtubules, and thick filaments, which are only found in muscle cells. And the cytoskeleton actually does look like a skeleton inside the cell. That's how it's got its name. You can see from these images, the image on the left, the microtubules have been dyed yellow, so you can see those throughout the cell. And in the picture on the right, the microfilaments have been dyed green. So microfilaments are the smallest of all of the cytoskeletal elements. They are made of a protein called actin. And again, we're going to talk a lot more about actin filaments or microfilaments when we get to the muscle chapter. Microfilaments are most common in the periphery of the cell. So in this image from your textbook, you can see them here around the edge of the cell, and they can extend up and form a microvilli as well. They function to anchor the cytoskeleton to integral proteins. So that allows the cell to basically have some stability and structure by having its insides anchored to the membrane. It also can determine the density and consistency of the cytoplasm. So the more microfilaments you have, the more dense and less flexible the cell would be. Actin can interact with a protein called myosin to produce movement or change cell shape. And this is a very key point to how muscle cells function, which we will see a little bit later in chapter 10. And microfilaments can also be called actin filaments or thin filaments. So you'll see all 3 names used interchangeably. And another key structure microfilaments form is a thing called terminal web, which is again shown in this image as this collection of microfilaments that run across the top portion of a cell. And this is found in cells that form a layer or a lining, and we call these types of cells epithelial cells, and we'll go into more detail about epithelial tissue in chapter 4. The next type of cytoskeletal element are intermediate filaments. As the name suggests, these are intermediate in size. The main protein you would find in an intermediate filament can vary from one cell type to another, but one example is that your skin cells contain intermediate filaments made out of a protein called keratin. The intermediate filaments help to strengthen the cell and to maintain its shape. It can also stabilize the position of the organelles inside the cell. It can stabilize the position of the cell with respect to surrounding cells. And it's one of the most durable of cytoskeletal elements. So because a lot of them are made of keratin, keratin is one of the most durable proteins. So to give you an example, your fingernails and hair are made out of keratin protein, and those can last long after the rest of the body has decayed. Microtubules are very large, hollow tubes, and they are made up of a protein called microtubulin. They are very dynamic, which means they're constantly being built, broken down, and rebuilt. And again, I have a really cool video I'm going to show you as part of the in-class activity that will help you visualize this. They function as the primary component of the cytoskeleton. They allow for strength and rigidity, and they can help to anchor the organelles. The disassembly and reassembly allows the cell to change its shape. So microtubules can be broken down at one end and built on the other end of the cell to propel the cell forward. They also, this is a really cool function, they serve as tracks, like train tracks for organelles and vesicles to move around the inside of the cell. And there are little proteins that we call molecular motors that actually carry vesicles and organelles just like their cargo. And they walk along these microtubule tracks. And 2 examples are proteins called kinesin and dynein. And again, I've got a really cool video to show you where it shows you actually what these proteins look like. And they do look like little guys carrying cargo. These molecular motors require ATP because, as you can imagine, moving stuff around the inside of the cell would be a very energy-consuming process. Microtubules also function and play a large role in cell division, and they are the structural component of some other organelles, as we'll see in a moment. Thick filaments are very massive bundles of subunits that are composed of a protein called myosin, and these are found only in muscle cells, and we're going to talk a lot more about these later in the semester. Here's a picture of a muscle cell, and again, this is coming in more detail in Chapter 10. The thick filaments are here, and then notice that microfilaments are called thin filaments here, but they are the same thing. And again, we're going to get into much more detail in Chapter 10. So that was the cytoskeletal elements. Now we're going to look at some other non-membranous organelles, starting with the microvilli. So microvilli are small projections of the plasma membrane. They greatly increase the surface area of a cell. So for example, you can find these lining your intestines, and it allows your intestinal cells to have a larger surface area for which that they can absorb nutrients from the food you eat. And they are made of a core of microfilaments. So the microfilaments, the same ones that we just talked about, actually form the core of the microvilli. The next structure are the centrioles. This is a structure made up of microtubules. It's only found in animal cells, and it has what we call a 9+0 arrangement of microtubules. And what that means, the 9 part refers to the 9 triplets that are found in a circle around a center that has no microtubules. So that's the 9+0. The centriole forms what's called the spindle apparatus when a cell divides during cell division. Some cells lack centrioles, like mature red blood cells, skeletal muscle cells, cardiac muscle cells, and most neurons. So what do you think it means if a cell has no centrioles? Well, since the centriole forms one of the main structures needed during cell division, it means that these cells do not divide. And then the centrosome is a region of cytoplasm that is found close to the nucleus, and it surrounds a pair of centrioles. So 2 of them together make up this area around the nucleus called the centrosome. Cilia are long, slender projections of the plasma membrane, and cilia are made up of microtubules. So if you remember, the centrioles had a 9+0 arrangement, whereas the cilia have a 9+2 arrangement. And what that means is that there are 9 pairs around the outside and then 2 microtubules in the middle. And a cilia, again, this is extending off of the plasma membrane, and it's anchored to the cell in the plasma membrane at the bottom by a structure called the basal body, which looks a lot like a centriole. This has the 9+0. Cilia can beat rhythmically in an oar-like motion, so imagine how an oar works in a canoe or a boat. So there's a power stroke and a return stroke, and it allows the cilium to actually move materials and in some cases even move the cell. So for example, you have cilia lining your trachea and some of your bronchi, and this helps to move mucus and phlegm up your throat. Cilia can also play an important role in signaling, and it's found on a lot of cells during the developmental process. The next structure is a ribosome. These are the protein factories of the cell. They are composed of 2 subunits, a large subunit and a small subunit. And the ribosomes are made of 60% rRNA, which actually stands for ribosomal RNA, and about 40% protein. Ribosomes alternate between a free state and a bound state. So if they're free, they're just floating around in the cytoplasm. And if they're bound, that means they're fixed or attached to the ER. And the ER is the endoplasmic reticulum. And again, ribosomes are the cell's protein factories. This is where you're going to make new proteins. And again, remember, proteins are the workhorses of the cell. So cells are always constantly making new proteins. And finally, we have the proteasomes. Proteasomes contain protein digesting enzymes called proteases. Anything that ends in an ase is an enzyme. That was one of your root words. So proteasomes, because they contain enzymes that can digest proteins, they are responsible for removing and recycling damaged, denatured, and abnormal proteins. So if a protein needs to be destroyed because it's been worn out and it's no longer useful, it's tagged with a little molecule called a ubiquitin. That's a signal that it needs to be thrown in the trash. The proteasome comes along, it engulfs it. The proteases inside will break it down into its different components, these smaller peptide units. These will at some point be broken down into amino acids, which will be used to make new proteins. In this section, we will talk about the membranous organelles, which include the ER, Golgi apparatus, lysosomes, peroxisomes, mitochondria, and the nucleus. So we'll start with the endoplasmic reticulum, or ER. This is a network of intracellular membranes, so it's very membranous, and it shares membranes with the plasma membrane, with the nucleus, and with the Golgi apparatus. So there are two types of ER, and the two types depend on whether or not ribosomes are attached. So there is a rough ER that has ribosomes attached to it, and a smooth ER where there are no ribosomes. So let's look at the smooth endoplasmic reticulum first. So no ribosomes are associated to this, so that's shown here on the right side of the screen. It's the portion without the ribosomes. Its function can depend on what type of cell it is found in. Some of the functions include making the phospholipids and the cholesterol for the cell membrane. All cells actually have this function. So this is one function that is common to smooth ER in all cells. And basically, this is where new plasma membrane is made. Then in your reproductive organs, the smooth ER functions to make steroid hormones like estrogen and testosterone. The ER also can synthesize and store glycerides, and you might have heard of one type of glyceride called a triglyceride. A glyceride is basically a stored form of fat, and so you would find a lot of smooth ER in liver cells and adipocytes that would be making the glycerides. The smooth ER can also synthesize and store glycogen. Glycogen is a stored form of carbohydrate. It's actually a stored form of glucose. So you would find this a lot happening in skeletal muscles and liver cells. The smooth ER can also absorb and store ions like calcium, and this plays a big role in the way that muscle cells and neurons work. Again, we'll see more about this later in the semester. And it can play a role in the detoxification and inactivation of drugs and toxins. And so smooth ER that are in the cells found in the liver and the kidney cells would be doing a lot of this role. So the rough ER is the next structure, and it's called the rough ER because it looks rough with the little ribosomes attached to it. And so when you're trying to remember the function of the rough ER, try to also remember what is the function of a ribosome. So if you remember, ribosomes make proteins. So guess what? The rough ER functions with making proteins. So the ribosomes actually make the proteins and insert them into the rough ER where the process continues. So once the protein is made, the rough ER will take the protein and make some adjustments to it. So for example, if it sticks a carbohydrate on it, it makes a glycoprotein. The proteins are then packaged and shipped, so that's another function of the rough ER. It takes those proteins, it packages them up into little membrane packets called vesicles, and it transports them out to other areas. And the ratio of how much rough ER and how much smooth ER you have varies by cell type. So if it's a cell that needs to make a lot of proteins for a particular function, that cell would have a lot of rough ER in it. But if it's a cell that needs to make a lot of steroid hormones, for example, that type of cell would have more smooth ER in it. So the ratio varies based on the cell's function in the body. The next organelle is the Golgi apparatus. This consists of flattened membranous sacs called cisternae. And this is also part of that protein making network, and as well as like the steroid hormone making network. So proteins can be sent to the Golgi apparatus where they are further modified and packaged, and they are again put into little vesicles where they can be released from the cell through a process called exocytosis. They can also handle hormones and other compounds in this manner as well. The Golgi also plays a role in renewing and modifying the cellular membrane. So again, it plays a role in shipping and packaging those phospholipids and cholesterol molecules. And it can also package special enzymes inside of vesicles that the cell can use for other functions. And we're going to talk about these in a minute. These are called lysosomes. And that brings us to lysosomes. So lysosomes are little vesicles that contain digestive enzymes. Think of these as the stomachs of the cell. They are produced by the Golgi apparatus, and they have a couple of functions. One of them is that they can fuse with damaged organelles to break them down. So the lysosomes contain actually hydrolytic enzymes. These are enzymes that break a lot of stuff down. Kind of think about like how the stomach acid in your stomach works. It's different compounds, but it's the same concept. So if the lysosome fuses with a damaged organelle, the damaged organelle will be broken down into its component parts, which can be reabsorbed and reused by the cell. Cells can also take in extracellular matter. This could be, something the cell needs to digest, or it could even be a bacteria, and that extracellular matter is brought into the cell through a process called endocytosis. Again, it can fuse with a lysosome, and the lysosome will break that foreign matter down into pieces, which can be reabsorbed or ejected as waste. And their final, and they can also function with cleanup and recycling. So again, they're like the stomach of the cell. They can digest and get rid of anything that you need them to. They can also do what's called autolysis. Autolysis is where the lysosome basically ruptures inside the cell, releases all of those hydrolytic enzymes, and those hydrolytic enzymes will proceed to break down everything in the cell. So we actually call this cellular suicide, and it has a name called apoptosis, and it's basically where the cell is killing itself. There are a whole host of diseases called lysosomal storage diseases that can have some very serious symptoms, and they result from lysosomes not being able to do their function. So lysosomes are very important for cells to operate correctly. The next organelle is a peroxisome. Just like the lysosomes, peroxisomes also contain dangerous chemical reactions that the cell wants to isolate inside this little organelle to keep away from the rest of the cell. These guys are smaller than a lysosome. The membrane is different from a lysosome, and these are produced by other peroxisomes dividing in two. They function to absorb and break down fatty acids, and they produce hydrogen peroxide as a byproduct, and that's why they're called peroxisomes. And hydrogen peroxide is a very destructive molecule. So that's why the cell wants to have these reactions take place inside this organelle to keep it away from the rest of the cell. And then an enzyme called catalase can come along and break down the hydrogen peroxide to oxygen and water, which are harmless, and then it is safe for the cell to be exposed to oxygen and water, but not to the hydrogen peroxide. The mitochondria are the energy producers of the cell. So the number of mitochondria you find in a cell will depend on the cell's energy requirement. So an adipose cell that's just sitting there storing fat may not have many mitochondria because it's not doing a whole lot, whereas a muscle cell may have tons of mitochondria because it's needing a whole bunch of ATP for muscle contraction. Mitochondria contain a double membrane. So the inside, so there's an outside membrane, and the membrane on the inside forms these folds called cristae, and then the very inner compartment inside that inner fold is called the matrix. And the function of the mitochondria is to produce ATP. So it is the energy factory of the cell. So the production of ATP could actually be a very detailed topic, but we're just going to quickly go through it in the barest form possible, because there could be like whole sections and classes that goes over this process. So this is all happening it starts in the cytoplasm, then it moves into the mitochondria. The first step is called glycolysis, and it actually does occur outside in the cytoplasm. And this is the first step where you take like a glucose molecule and break it down into pyruvate, and it does not require oxygen. The second step is that the pyruvate goes into the mitochondria and it enters this reaction cycle called the citric acid cycle. This citric acid cycle, we say it indirectly requires oxygen because it won't happen if oxygen is not present, although the oxygen itself is not used here in the citric acid cycle, but it still won't happen if there is no oxygen. And the citric acid cycle produces carbon dioxide as a byproduct, and this is very important later in AMP2 when we get into the entire function of the respiratory system. The entire reason you have a respiratory system is to get oxygen to the mitochondria and to get rid of the CO2 that was produced as a waste product, right? So if you think about it, the whole reason we have a respiratory system is all down to keeping our mitochondria and all of our billions of cells happy. After the citric acid cycle, there is the electron transport chain. And this does directly require oxygen, and this final step is where you're going to produce your new ATP molecule. So when you do make ATP in the presence of oxygen, we call that aerobic metabolism or cellular respiration. There is also a thing called anaerobic metabolism where you can make ATP without oxygen, but most of the cells in your body cannot do this, with the exception of muscle cells. Muscle cells are the only cells that have the capability to make ATP without oxygen. So aerobic metabolism is making 95% of the ATP that is needed by the cell. So again, this is a very important process. This is making the ATP, which we talked about in chapter 2, is the energy currency it's needed by the cell to do all of its jobs. And the last membranous organelle is the nucleus. This is the control center for all cellular operations, so you can think of it as like the brain of the cell. It contains the information necessary to make over 100,000 different proteins, and this is just in the human body, right? So all cells have DNA, but in the human body, we know that the DNA has directions for at least 100,000 different proteins. Most cells only have one nucleus. There are some exceptions. Skeletal muscle cells can have multiple nuclei, sometimes up to 100 nuclei in one cell. And then mature red blood cells actually do not have a nucleus. The nucleus's structure is a double membrane, so it's surrounded by this double membrane. It's called the nuclear envelope. The paranuclear space is the space between the two membranes. And then there are little holes that allow stuff to come in and out of the nucleus, and those are called nuclear pores. Inside the nucleus, the fluid contents is called the nucleoplasm. There are a network of fine filaments inside the nucleus called the nuclear matrix. And then there is this darker area inside the nucleus called the nucleolus, or nucleoli is the plural. And this is the area where you make new rRNA, which is ribosomal RNA, and you also make the ribosomal subunits. Remember that ribosomes have a large subunit and a small subunit. The nucleus can also contain ions, enzymes, and of course RNA and DNA. So the nucleus, one of its primary functions is to house and contain the nucleic acids. Now we will talk about how DNA actually controls the protein synthesis. And there are a lot of details here. I'm going to try to only point out the most relevant details, and hopefully this is material that you've had previously in an intro bio class. So DNA, we've already learned, is located in the nucleus, and in the nucleus the DNA can be found wound around proteins called histones, and so winding the DNA around the proteins helps to stabilize it and keep it organized. Then you have these structures called nucleosomes, which are multiple complexes of DNA and histone proteins. And then from the nucleosomes, there are different levels of coiling, so how tightly the DNA and the histones coil together. If it's just loosely coiled together, we call that chromatin, and that is typically how you will find the DNA in a cell that is not currently undergoing cell division. However, the DNA can be supercoiled or very tightly coiled, and when this happens, the DNA is actually visible under a microscope in a structure called a chromosome. And you typically only see these when a cell is actively undergoing cell division. And just information to have, humans have 23 pairs of chromosomes. You get one pair from each parent, your mother and your father, so you have a total of 46 chromosomes. So let's talk about how DNA stores information. So DNA has specific sequences within it called genes, and genes are the functional units of heredity. This is what we pass on from parent to offspring. And so if we look at the little picture down here, we see the chromosome, which has, if you take the proteins out, you've got the DNA, and then genes are specific regions on the DNA that have the instructions to make a specific protein. So genes contain the entire set of instructions to make a specific protein. In order for the gene to be read, it follows what's called the genetic code, and that is the information is stored in a triplet code. Triplet means 3. So there are these triplets of 3 nitrogenous bases, and remember we talked about the nitrogenous bases in chapter 2, and in this picture here, our example, we've got guanine, cytosine, and adenine. So 3 bases together make up a structure called a codon, and that is going to be information on how to add one amino acid to a protein. So with all of the letters of DNA, there are 64 possible triplets, and these can code for all 20 amino acids. So there is some repetition where some codons can code for the same amino acid. The number of triplets, or the number of codons you have in a gene, depends on the total number of amino acids in the protein. So you have short proteins and you have long proteins. So a short protein would have a short gene, a long protein would have a longer gene. And this image here just reinforces that you have the codons in the DNA, which are the sets of 3 nucleotides. These get transferred into RNA, as we'll see in a moment, and then that information is then used to build a protein 1 amino acid at a time. Interestingly, only 2% of our DNA actually contains genes. The rest of the DNA has code for making tRNA and rRNA, which both participate in the protein synthesis process. There are some of these areas of DNA that contain regulatory sequences that determine when genes are turned on, when genes are turned off. And then there are some areas that have no apparent function. Mutations can rapidly accumulate in these areas, and repeating sequences found in these areas can actually help identify a unique individual. And these are the areas they use with like DNA fingerprinting when they're trying to identify a suspect who might have committed a crime. It's quite astounding to think that your DNA is actually 99.9% similar to another person's. So it's only that 1% difference that allows us to do DNA fingerprinting, but it also points to just how much DNA we have, that the 0.1% is enough to tell one person from another. So how are the proteins actually made? Well, remember the gene has the information for making the protein, and so there's a term called gene activation, and this refers to when the cell decides it needs a particular protein, then that gene is turned on. So there are regulatory sequences that say when and where we should make a particular protein. And a promoter is a name that we use for the control segment at the beginning of each gene, and things can bind there to turn the gene on or off. So when it's time to make a protein, the first step is transcription. So an enzyme called RNA polymerase binds to the promoter region, and it will transcribe the DNA into mRNA. So when you're trying to remember the difference between transcription and translation, think about transcription as being taking something from one language and keeping it in the same language. For example, in doctors' offices, often doctors will transcribe or they'll record their notes, and then someone will later listen to their notes and type them out. So we're going from like English to English. So transcription is the same language to the same language. And in this case, when I say same language, I'm talking about nucleic acids. So remember in chapter 2, both DNA and RNA are nucleic acids. So it's essentially the same language. And remember, when you're trying to differentiate between some of these enzymes, RNA polymerase makes RNA. So DNA is double-stranded, as we've mentioned before. So when it comes to genes, there is always one strand called the coding strand and one strand called the template strand. And RNA polymerase will read the template strand to make the mRNA. So the mRNA ends up being identical to the coding strand, except remember that mRNA has U's, uracil, whereas DNA has T-thymines. Otherwise, the mRNA is identical to the coding strand of DNA, and it's made by reading the template strand. So this is a summary of the transcription process. So we start with the double-stranded DNA on the left. The two strands are peeled apart. We have RNA polymerase comes in and it binds to the regulatory sequence at the beginning of the gene. It reads the different nucleotides all the way down. As it reads them, it adds the complementary RNA nucleotides. So remember, we said G's go with C's, A's go with U's, T's go with A's, etc. And then when it's done, the two DNA strands go back together, and now we have an mRNA strand, a messenger RNA strand. So after transcription is finished, we have this pre-mRNA. So it's an mRNA strand, but it's not quite finished yet. There is some editing that happens before it leaves the nucleus. There are structures called introns that get cut out, and structures called exons that are spliced together. There are also some other modifications that you would have to learn if this was a cell bio class, which it's not. So in this picture here, let's look at the gene on the top. So the introns would be represented by the green areas, or in the mRNA, it's just the lines down here. And the exons are the ones that are in red. So we've got 1, 2, 3, 4 exons. So another thing that can happen, in addition to just cutting out those introns and putting together the exons, is that you can also have what's called alternative splicing, which means that in some cases, even different exons might get cut out. So in this example on protein A on the left, we have exons 1, 2, and 3, but we actually cut and got rid of 4. And then maybe, in another situation, we make a protein out of exons 1, 2, and 4, where we cut and got rid of 3. This is why we only have 20,000 genes, but we can make over 100,000 different proteins. And the reason is you can use the instructions in a gene to make more than one protein by doing this alternate choosing of exons from within the gene. Then once you have your final mRNA with the final instructions all together, that leaves the nucleus through the nuclear pores. The next step is translation. So think about what translation means. Translation means going from one language to a different language. So you translate something from English to Spanish, for example. So in this case, the mRNA strand, which is a nucleic acid, is used to form a protein, which is a chain of amino acids. So it's two different languages. We're going from the nucleotide, nucleic acid language to the protein amino acid language. The key players here are the ribosome, which we've already mentioned are the protein factories of the cell. And we've already talked about transfer tRNA in chapter 2. This is the RNA that delivers amino acids to the ribosome. And tRNA molecules have anticodons that match the codons on the mRNA. So we already mentioned how the genes are broken into triplets called codons. Those get copied into the mRNA, and then the tRNA can have matching ones that allows them to build the correct amino acids into the protein. So here's a summary of how translation works. So we start up here in the top left, we got mRNA coming out of a nuclear pore of the nucleus. Then when it gets out into the cytoplasm, it will bind to a ribosomal subunit. It actually binds to the small subunit. You've got a tRNA that comes in to help. Then you get the large subunit and everything's set up, and then you have tRNAs that come in, read the code, and add the correct next amino acid. And this continues over and over again until you have a completed protein, which is a string of amino acids, also called a polypeptide. So here's an overview of protein synthesis from start to finish. So remember that protein synthesis begins in the nucleus. The information is in the DNA. The DNA is going to put it into an mRNA strand through the process of transcription. The mRNA strand undergoes some modifications, and then it leaves the nucleus through the nuclear pore. Once it is outside of the nucleus, it can either go to a free ribosome to have a protein made, and in this case the protein just stays in the cytoplasm, or it can go to a bound ribosome, which is a ribosome attached to the rough ER. And in this case, the protein is inserted into the rough endoplasmic reticulum, where it undergoes further modifications. This is where we might stick a sugar on it to make a glycoprotein. Then the rough ER is going to package it into little vesicles. Those little vesicles are going to travel to the Golgi apparatus. It will then fuse and become part of the Golgi apparatus, where it could undergo further modifications and changes. Then it is packaged again. And in some cases, it is packaged and secreted to the outside of the cell. For example, insulin is a protein, and with this pathway, you would secrete insulin to the outside of the cell so it could go into the bloodstream. And sometimes the proteins are used, the digestive proteins are used to make new lysosomes. So that is an overview of the whole protein synthesis process. So you can really think of cells as little factories. Right, we've got the cell membrane, which controls what things enter and can exit the cell. So it can let in the correct nutrients, it can let metabolic waste leave. And then inside the cell, we've got the brain of the nucleus that has the instructions. Those instructions are passed to the other parts of the factory through RNA. And then we can make things like the ribosomes can help with making the proteins. And in the rough ER, we can take those proteins, we can fix them up and add a little bit extra to them. Then they go to the Golgi, where they're further packaged and they're exported to outside the cell. Some of those proteins may be used for cell repair. And then down here, we've got our energy generator, which is our mitochondria making our ATP. So the final part of this section, how does DNA control the cell structure and function? Right, so we've talked about all of these different organelles, all of the different things that cells can do, and I just gave you a rather simplistic view of how DNA makes mRNA, which makes protein. And you're like, well, whoa, how do we get to this final complex cell just from this DNA to RNA to protein? Well, there's the direct control, where the DNA has the instructions for making structural proteins. So it's making the proteins that, say, for example, form the cytoskeleton. It's making the proteins that are inserted into the membrane. So remember those integral and peripheral proteins. And it's making proteins like the secretory proteins, like the insulin I mentioned, that gets secreted to the outside of the cell. But there is also a lot of indirect control. The DNA also has the instructions for making enzymes. And enzymes catalyze a whole lot of different reactions in and out of the cell. So DNA has the instructions to make the enzymes, and then the enzymes go out and do further work in the cell. So in this case, DNA is basically making the tools that then go out and do more jobs. And with these two things together, you end up with an entire cell that does all the functions it needs to. Now here's something to think about. All the cells in your body have the same DNA. The cells in your eye have the same DNA as the cells in your finger, have the same DNA as the cells in your brain, have the same DNA as the cells in your stomach. So, why is a neuron, which is a nerve cell, so different from a skin cell if they all have the same master instructions? And the answer goes back to that gene activation. We also call this gene expression. So remember I said that you can turn genes on or off. So they have like these little regulatory sequences that allow you to switch them on or off. Well, the reason we can have so many different cell types in our body, so we can have neurons, we can have skin cells, we can have liver cells, we can have kidney cells, blood cells, is because in each of these different cell types, different genes are turned on and off. So basically they all have the same master instruction handbook, but they're reading from different pages. So the neurons may be reading from pages 100 to 300, whereas your skin cell may be reading from pages 800 to 900. So they're reading different sections of the master instruction manual. And the term called differentiation refers to the development of the specific cellular features as a result of turning on or off different sets of genes. And this happens during development. So we all start from a single cell when the sperm joins with the egg during conception, and we have a zygote. And then as the embryo progresses, that cell is dividing over and over and over again until it's making hundreds and billions and tons of cells. And then as it makes all of those different cells, different areas of those cells start to differentiate, which means they turn on specific sets of genes. And so this group of cells will become your brain cells, another group of cells will become your stomach cells, a further group of cells will become your liver cells, etc. Now we will talk about diffusion and osmosis, which is the first part of our coverage of cellular transport. So remember that the term permeability means the property of a membrane that determines what substances can pass through it. And so you can range from freely permeable, which means anything can pass through the membrane, to impermeable, which means nothing can pass through it. And the plasma membrane falls somewhere in the middle. So we say that the plasma membrane is selectively permeable. And what that means is the permeability can actually differ from cell to cell, and it can depend on what proteins and lipids are actually found in the plasma membrane of that particular cell type. So remember, the proteins can form channels and carrier proteins, and that controls what is allowed into and out of the cell. There are two broad classes of transport. Passive transport is when you move stuff without the expenditure of energy, so it happens with no energy consumption whatsoever. And active transport means that you require energy to make it happen, and that energy is usually in the form of ATP. So diffusion is the movement of a substance from an area of high concentration to an area of low concentration of that substance. It ignores any other substances around it, and it only moves from its own area of high to low. So keep in mind, diffusion goes from an area of high concentration to an area of low concentration. The term concentration gradient refers to a difference in concentration. So if you look at the image here, where we drop a little cube of like beef bouillon or something into some water, and so the highest concentration in the beginning would be right around the cube that you dropped into the water. And then up here at the top, you would have a very low concentration. So the difference is what we call a gradient. And diffusion works to eliminate or get rid of that gradient. So the molecules will diffuse out from the area of high concentration to the area of low concentration until eventually the entire concentration is the same and there is no longer any gradient. One example is carbon dioxide. So you have a high level of CO2 inside your cells. Remember, it is being produced as a byproduct by the mitochondria when they make ATP. There are lower levels in the interstitial fluids, which is the fluid that is found in your tissues. and the lowest levels are in your actual bloodstream. So this means that carbon dioxide can move down its concentration gradient, so it goes from the cell into the interstitial fluids and then into the bloodstream. And so that way the bloodstream can then carry it to the lungs, where it can be exhaled through your lungs. So again, this is diffusion, and when you have diffusion, I should also point out diffusion is passive, so this does not require any energy. So think of it like sliding down a slide, does not require energy. So factors that affect diffusion: the distance that something has to travel. So if it's a shorter distance, it will happen faster. The size of the molecule, smaller molecules can move faster. The temperature, so if the temperature is higher, you get faster diffusion. Think about when you want to dissolve sugar in some sugar water. If you have warm water, it will dissolve the sugar faster than cold water. So that's an example of how a higher temperature can help with a faster diffusion. And then a concentration gradient: the steeper the gradient, the faster the diffusion. And I know that sometimes students have trouble visualizing gradients, so I put a little chart here. So if a gradient, again, remember, is the difference between the area of high and the area of low. So when we talk about a steep gradient, think of it like a slide. This would be a very steep slide. And then over here, we still have an area of high and an area of low, but they're not as different from each other. So the gradient is more shallow. And just like with a slide on a playground, you'll go down the steeper one faster than you'll go down the more shallow one. So the steeper the gradient, the faster the diffusion. And then electrical forces also play a role, because we have a lot of ions in our bodies. So like sodium ions, potassium ions, calcium ions. And with ions, you have charges, positive and negative charges. And so the diffusion of these ions will depend on the electrical forces surrounding them. Remember that opposite charges attract. So a positive charge is attracted to a negative charge. So another thing to keep in mind, and we talked a little bit earlier about differences between the inside of the cell and the outside of the cell. And one of those differences is actually charge. So cells have a slight negative charge on the inside, and there is more of a positive charge on the outside of the cell. So this can affect the way that ions in the body diffuse. So when you're looking at the diffusion of ions, you can't just look at the concentration gradient, which is what we call a chemical gradient, but you also have to look at the electrical gradient, which is the difference in charges. So if you combine the chemical gradient, again, which is concentration, and the electrical gradient together, we call that the electrochemical gradient. So here's an example. So we mentioned earlier, sodium concentration is higher on the outside of the cell. Sodium concentration is lower on the inside of the cell. And again, remember, that's an important thing to cement in your brain. It's going to come back later this semester. And something I didn't mention earlier is that chloride ions are also higher outside the cell and lower inside the cell. So if we talk about diffusion of sodium ions and the diffusion of chloride ions, sodium is going to diffuse faster because it has both the concentration gradient and the charge in its favor. So it will want to go from high to low, right? So high outside, low inside. So sodium will want to diffuse into the cell. And then the charges are in its favor as well. Sodium is positive and the inside of the cell is negative. Positive likes negative, so again, it's in its favor to come into the cell. However, you would have a slower diffusion of chloride, because whereas you have a high to low concentration, now you've got a negative ion going into an area that is negative, and like charges repel, not attract. So chloride has only one of the two gradients in its favor, and so therefore it would have a slower diffusion than the sodium. So of course we're talking about cells, so we're ultimately talking about diffusion across the plasma membrane, and molecules can diffuse across the plasma membrane in only one of two ways. The first way is that they can go directly across the phospholipid bilayer, and the second way is that they can pass through a membrane channel protein, which are those integral proteins stuck into the phospholipid bilayer. So the first option, this is the option where it goes directly through the membrane. That is called simple diffusion. Passage goes directly through the phospholipid bilayer. However, remember the bilayer was a little sandwich with a hydrophobic middle, and so only small hydrophobic substances can do this. They're the only ones that can cross that middle hydrophobic region. So things like alcohol, fatty acids, steroids, oxygen, carbon dioxide, and certain lipid-soluble drugs are able to pass through the membrane directly. Everything else needs channels or carrier proteins, but for diffusion, facilitated diffusion, this is only going to happen through channel proteins, so we call this channel-mediated diffusion, and ions and other small substances pass through a membrane channel protein, which is one of these transmembrane integral proteins. As I mentioned earlier when we were talking about functions of membrane proteins, these channels are very specific for the type of molecules they let pass, so a sodium channel will not let anything but sodium pass through it. These also only allow very small solutes to pass, so you actually have to have other proteins called carrier proteins to allow larger molecules, like say insulin, to pass through. So there are multiple types of channels. A leak channel is a channel that is always open. Because it is always open, ions are able to passively diffuse through it all of the time. Now, contrast that with gated channels, which we're going to talk about later in the semester. These channels have different kinds of gates that can open and close to control when ions pass through them. All right, so now let's talk about osmosis, which is actually the diffusion of water. So if a solute cannot cross the membrane to equalize the concentration, because remember we just said that like the plasma membrane can control which solutes can cross it by the presence of channels. So if a membrane does not have a channel for a particular solute, that solute cannot cross the membrane. So if solutes can't cross, then water can cross to equalize the overall solute concentration. Notice I put this in bold italic. When water moves to equalize concentrations, it doesn't care about individual solutes. It is looking at the overall solute concentration. And the term osmosis, again, this is the diffusion of water. And it technically is the movement of water from a high water concentration to a low water concentration. But you need to start thinking of osmosis in the terms of solute concentration, because that is what is going to help you in many different chapters to come in A and P. So think of osmosis as water moving from an area that has low solute concentration to an area that has high solute concentration. Another way to think of it is water is moving to dilute the highly concentrated solute. So again, the goal is to equalize the overall concentrations. It doesn't look at individual solutes. It is trying to equalize the overall concentration. So here is the picture from your textbook. We have a bunch of purple dots which represent the solute molecules. The water molecules are shown by the little blue dots. And we have a membrane here that does not let the pink solute molecules pass. So there are more pink solute molecules over here on the B side than there are on the A side. So if the pink molecules could pass the membrane, they would diffuse themselves and make themselves equal. But in this case, we have a selectively permeable membrane that does not allow the pink dots to move, so water has to move instead. And water will move toward the area that has the highest solute concentration. So water will move from side A to side B until the two concentrations are equal. Water crosses the plasma membrane through channels called aquaporins. So water can always cross the plasma membranes to equalize total solute concentration. So another term to be familiar with is osmotic pressure. This is the force that water moves with. All right, so we just talked about how water will move to equalize the concentration. So water will move toward an area of higher solute concentration, and it actually will exert a pressure or a force to do this. So you can use a little U-shaped tube like this and actually see one side of the water get higher on one side than the other, which means water is going against gravity to do this. So that tells you the water is exerting a force. So you can measure that force, and if you measure that force, that is what we call osmotic pressure. And the reason this is important is because this is a force that is very important for a lot of your body's physiological processes, like the way blood and fluid in blood moves in and out of the blood to get to the tissues, and also the way your kidney works in filtering the blood. So having this osmotic force or osmotic pressure is very important in a lot of physiological processes. Another term to be familiar with is osmolarity. Osmolarity is just the measure of the total solute concentration. Again, we're not looking at individual solutes. We're not looking at sodium and chloride and calcium. We're looking at all of them together. So you should also be familiar with tonicity, which is a measure of the relative solute concentration between two solutions. So these involve some terms like isotonic, which means two solutions have the same solute concentration, hypertonic, which means one solution has more solute concentration than another, and hypotonic, which means one solution has less solute concentration than another. These are your root words. Hyper means more, hypo means less, iso means the same. Now notice as I was describing those that I kept saying, you know, one has more solute than another. One has the same solute concentration as another. These terms only make sense if you are comparing two solutions. You can't set a glass of water on a counter and say, this is a hypertonic solution. That would make absolutely no sense because you have to be comparing it to something else. These are comparable terms. You are saying solution A has more or less or the same solute as solution B. So for example, here are 4 different solutions. We have solution A, B, C, and D. The little dark blue dots are the solutes. So we could say, for example, we could say D is hypertonic when compared to B. So again, what we're saying is D has more solute than B. We could also say C is hypotonic compared to B. Or we could say A is isotonic to B. But again, you only use these terms when you are comparing different solutions. Keep in mind, if the solute can't diffuse, then water is going to move through osmosis from the hypotonic area to the hypertonic area. In other words, just drill into your brain, water moves towards the highest solute. Water moves towards the hypertonic area. So if I were to put C and D right next to each other, and I were to stick a membrane there so that water can pass, but the solute can't, what's going to happen is water is going to go from C to D. So water is going to move toward the area that has the most solute. And again, think of it as the water is trying to water is trying to dilute the concentration over here and make it the same as C. So why do we care about this? Well, let's talk about how this works with cells. So if you take a cell, and this is an example showing a blood cell, if you take a blood cell and you stick it in an isotonic solution, and again, keep in mind when I say an isotonic solution, I'm comparing it to something, that means I'm comparing it to the cell. So I'm saying the cell is isotonic compared to the solution. But what that means is concentrations are the same, so water molecules are going to move in as much inside as they do outside, and everyone's happy. If you stick a red blood cell into a hypotonic solution, again, remember, if I'm saying that the solution is hypotonic, then by default, that means the cell is hypertonic by comparison. Because you can't just have something hypotonic without something also being hypertonic. Because if you're saying one has less solute than another, then the other thing has to have more solute. So keep in mind that we're saying that the cell is hypertonic in this case. And remember what I told you about water always moves toward the hypertonic solution. So water would move into the cell, and it would cause the cell to swell up and potentially burst open. And then finally, if we put a cell into a hypertonic solution, again, so what does that mean? If the cell is in a hypertonic solution, then that means the cell itself is hypotonic by comparison. And again, remember, water wants to move toward the hypertonic region. So in this case, water will move out of the cell into the solution, and the cell will shrivel up, which is what we call crenation. So some terms to know: hemolysis is when a red blood cell bursts from absorbing too much water. So that would be the second case here. When you have a red blood cell in hypotonic solution, water goes inside, it swells up, it can burst open, and that's hemolysis. Crenation is the example shown in C. That is when a cell shrivels up and shrinks from losing water. And why do we care about this? Well, they use the example of blood cells here because this comes into play when you're giving someone an IV, like a saline IV. You would never give someone an IV of like pure water. If you were to give someone an IV of pure water, that's the same as injecting hypotonic solution, right? The water has less solute in it than your red blood cells. So if you injected someone or gave someone an IV of water, basically their red blood cells would swell up and burst. And as you can imagine, that's not something you want to happen. Same thing if you give them a solution that has too much solute in it. So a lot of our IVs that you get when you go to the hospital are called normal saline, and the concentration is 0.9 grams per deciliter, and that is an isotonic solution compared to our red blood cells. So when you get an IV drip of normal saline, it is not going to disturb your red blood cells because it is isotonic. So now we've got to transport part 2, and in this section we're going to cover carrier-mediated transport and vesicular transport. So as I mentioned previously, the channels only allow very small solute molecules to pass through, so larger molecules require carrier proteins to shuttle them across the plasma membrane. So all carriers are specific to the type of solute that they can transport. They have saturation limits, which means they can only transport so many molecules like per minute or per second. So they have a high limit to how fast they can work. And they are tightly regulated. So other molecules can bind to them to turn them on and off and to tell them when to bring things in and when to hold off. So a term you should know is co-transport. With co-transport, two different solutes are carried in the same direction simultaneously, and that's shown in the second image here, where the blue and the purple molecule are carried both from one side to the other at the same time. That's also sometimes called symport. And counter-transport is when one substance is moved into the cell while one is moved out of the cell. So in the third picture, we've still got the blue and purple molecules, but now they are being moved in opposite directions. The blue is being taken out, the purple is being brought in, and that's also called antiport. So facilitated diffusion, we already mentioned, is a type of diffusion that can involve channel proteins, but it can also involve carrier proteins. And again, it's passive, because solutes are moving down their concentration gradient from high to low. And in these cases, the difference between the channel and the carrier is with the channel, the solute can just go straight through the channel. It's like a little protein hole in the membrane. But with the carrier proteins, a molecule has to bind to the protein. It causes the protein to change shape, and then the molecule can exit on the other side. So in this example, we're taking glucose molecules from outside the cell and we're bringing them inside. Glucose requires a carrier protein because it's too large for a channel. So it comes in, it binds to the carrier protein changes shape, and glucose comes out onto the inside of the cytoplasm. It still doesn't require any energy because we're still going from an area of high concentration to an area of low concentration. I mentioned on the previous slide that carrier proteins can be regulated, and one example is that glucose, the carrier protein, is regulated by insulin. So when insulin is present, it binds to these carrier proteins and allows them to work. And if insulin is not present, then the carrier protein just sits there and it won't function. So active carrier-mediated transport, or active transport, is when we are using energy to move a solute against its concentration gradient. So now we are using, we are moving the solute from an area of low concentration to an area of high concentration. So we're pushing it up the slide, so we're going against the gradient. And high energy bonds are used to allow these carriers to change their shape. And remember, our most common high energy bond comes from ATP. So some examples of active transports are ion pumps. These actively transport ions like sodium, potassium, calcium, magnesium, iodine, chloride, and iron. And again, it's moving them against its gradient. And if you have a particular pump that is moving two ions in opposite directions, so remember, if two solutes are going in an opposite direction, we call that countertransport. If this happens to be a pump, an ion pump doing it, we call it an exchange pump. And there's a very important one that we're about to go into detail with, and that is the sodium-potassium exchange pump. So remember back from the beginning of the lecture, I told you that it was very important to remember the differences in sodium and potassium concentrations from the inside of the cell to the outside of the cell. So as I mentioned in the beginning, sodium is high on the outside and low on the inside, and potassium is the opposite. Potassium concentration is high on the inside of the cell and low on the outside. It's very important for you to remember this. Homeostasis depends on this gradient. If your cells lost this gradient, you would die. This gradient is essential for your muscle cells to work and for your nerve cells to work. So your heart and your brain would both shut down without this gradient. So what happens if we stuck in leak channels? Remember, a leak channel allows passive diffusion from high to low. So if we stuck in leak channels, eventually the sodium would all move onto the inside until it was equal, and the potassium would all move to the outside until it was equal. But we don't ever want the concentrations of these two molecules to be equal. We want to keep it high sodium on the outside and high potassium on the inside. Again, this is very important for homeostasis of the human body. So to ensure that these gradients always stay the same, we have a sodium-potassium exchange pump. And this pump uses ATP. And for every one ATP molecule, it pumps out 3 sodiums and it pumps in 2 potassiums. So again, it requires energy because we are going from low to high. So we're going against the gradient for the sodium, and we're going from low to high against the gradient for potassium. Now, I have a video that I made for my lab course. I'm going to show you a little clip from it here, but I will also post the link to my website if any of you want to watch the full video to help you with your PhysioX activity in lab.
There is one particular pump that you should know specifically how it works, and that is the sodium-potassium exchange pump. First, you need to recall that the outside of the cell has the highest sodium ion concentration, and the inside of the cell has the highest potassium ion concentration. So the sodium-potassium pump uses one ATP molecule to move 3 sodium ions to the outside, so again moving it from low to high, and to bring 2 potassium ions into the cell, again moving them from low to high. So again, you should know that for each ATP molecule, 3 sodiums are pumped out of the cell and 2 potassiums are pumped into the cell.
And again, I will post a link to that video, and that may help you with your lab course. All right, so back to the sodium-potassium exchange pump. It can also be called the sodium-potassium ATPase, so you can see either term used. But one key thing to note about this, and this is why I'm stressing it so much, not only is the sodium and potassium concentration differences so essential to homeostasis, but this should tell you how important this pump is and how important it is to maintain these gradients. Up to 40% of the ATP that is produced by the mitochondria in a cell is used by this pump. To think of it in another way, up to 40% of the calories you are eating are going just to maintain your sodium potassium exchange pumps. So that right there should tell you how important they are and how essential it is to keep this concentration gradient. So there is also a type of transport called secondary active transport. In this instance, the mechanism itself doesn't require ATP, but ATP is required at a previous stage to maintain a concentration gradient of one of the co-transporters. So in this example here, we have a glucose molecule that we want to transport from low to high. So remember, normally that would be an energy requiring activity. But if we pair it with a sodium ion, remember we just got done saying sodium is high on the outside and low on the inside, so sodium can diffuse. And so as sodium is diffusing, which doesn't require energy, the glucose molecule can like hitch a ride and allow itself to go from low to high. So with secondary active transport, one solute is diffusing down its concentration gradient. In this example, that would be sodium. The other solute comes along for a free ride and basically gets to move from low to high without using energy. The energy was used earlier to make the high concentration of sodium on the outside. So we go back to our good old sodium-potassium pump, which is using 1 ATP to pump out 3 sodiums and pump in 2 potassiums. So here's where we use the energy to make the sodium gradient, and then we can use that sodium gradient to get a free ride of glucose. So again, this is another reason why we have to maintain this pump, because sodium can be used frequently as a co-transporter to move other things around without having to pay an additional ATP cost. All right, and the last part of the transport section is about vesicular transport. So vesicles can be used to transport materials in and out of a cell. This is also called bulk transport, and this always requires ATP. So anytime you're moving vesicles around, ATP is involved. So endocytosis is the process of bringing extracellular material into the cell. There are 3 types, and the first one is called penocytosis, and that is what this image up here is showing you. Penocytosis, which actually means cell drinking, is when the plasma membrane just pinches off a little bit of the extracellular environment and brings it into the cell. So it's non-selective. Basically, the vesicle is just scooping up whatever fluid is immediately surrounding the cell and bringing it inside the cell. Another type is phagocytosis, or cell eating. This allows the cell to capture much larger objects. So penocytosis is mainly for getting very small solutes. Phagocytosis lets the cell get larger objects, including things like bacteria that have made its way into the body. There's little pseudopodia, which are extensions of the plasma membrane that come out from the cell, wrap around the large object, and then the vesicle pinches off, and now you have what's called a phagosome, which is that vesicle that contains the large object. This will then fuse with a lysosome. Remember, lysosomes are the digestive stomachs of the cell, and they have the digestive enzymes in them. These will fuse with the vesicle holding the object, and the lysosome enzymes will break it down. So macrophages, which are a type of white blood cell, are cells that specialize in phagocytosis, and they specialize in actually cleaning up bacteria and other debris around the body. And I have a quick video here to show you of a white blood cell phagocytizing a bacterium. Okay, so here we go. So here is a big phagocyte, right? And it's chasing this little black bacteria. These are red blood cells. So this is a bacteria that's in the bloodstream, and you can see he's chasing him to eat him. And eventually he's going to catch up to him, and boom, phagocytosis. So he just used phagocytosis to bring that little bacterium into the main cell body so it can be digested. So that was phagocytosis. And the last type of endocytosis is receptor-mediated endocytosis, and for that we're going to this slide. So with receptor-mediated endocytosis, this is a highly selective process. So remember, like with the penocytosis, the vesicle just pinches off and grabs whatever is floating around outside the cell. But with receptor-mediated endocytosis, the cell specifically wants to bring in some type of specific solute. So this allows the cell to collect a specific target molecule in high concentrations. And how this works is that you actually have receptors, which if you remember is a type of membrane protein, and the receptors bind to specific ligands, and then those ligands and receptors then pinch off and form this vesicle that it can also be called a coated vesicle. And it's called a coated vesicle because it's coated all around the outside by these receptors. And then once this coated vessel comes in, now it has a very high concentration of the specific molecule. It can fuse with a lysosome, and the lysosome can allow the ligands to be removed, so taken off of the receptors, and that way the cell can get the ligands, and then the receptors can be reused and go back up to the plasma membrane for another grab, so that the cell can get even more of that type of specific molecule. And then the last type of vesicular transport is exocytosis. With exocytosis, the cell is now secreting stuff to the extracellular environment. So again, think of your root words. Endocytosis is bringing into the cell. Exocytosis is releasing to the outside of the cell. So exocytosis is the opposite mechanism as endocytosis. Ejected material, so material that's released, could be secretory products like hormones and proteins. We already saw an example of this when we looked at the entire protein making pathway. And the cell can also get rid of waste products this way. Sometimes endocytosis and exocytosis can be occurring in the same cell at the same time. And a really good example of this is if we look at an endothelial cell lining a blood vessel. So this blue cell in the middle here, this is all one cell. There's the nucleus right there in the middle. And so this is a cell that is actually lining the bloodstream, and we'll talk about endothelial cell linings in the next chapter. But we've got the bloodstream up here on one side of the cell, and the other tissues down here at the bottom on the other side of the cell. And so what we've got going on here is this cell is using endocytosis to grab stuff from the bloodstream. So it's using pinocytosis. Remember, that's where it just pinches off and gets bit of whatever was on the outside. So it's using pinocytosis to just pinch off bits of stuff that was in the bloodstream. Then those vesicles shuttle across to the other side of the cell, and then they fuse with the plasma membrane on this side and release those contents out into the surrounding tissues. And by this way, the endothelial cells can help cells that are not as close to the bloodstream still have access to the nutrients and gases that are available in the bloodstream. So this is a transport summary. So on the left side, we have our passive processes. Remember, passive means no energy is required. And so this includes diffusion, which is a solute moving from high to low concentration. We've got osmosis, which is water moving when the solute can't, and water always moves toward the hypertonic area, or the area with the highest solute concentration. And then we have facilitated diffusion, which is diffusion with the help of a membrane protein, and that can be either a carrier or a channel. On the right side, we have our active processes. All of these require ATP. So we have our active transport, and we looked at the example of the sodium-potassium exchange pump. And then we have our two types of vesicular transport. We've got endocytosis, and there are three subtypes: receptor-mediated endocytosis, pinocytosis, and phagocytosis. And then we have exocytosis, which is getting rid of stuff from the cell. We have a short little section on membrane potential. So if you remember, we mentioned earlier that the inside of the cell has more negative charges than the outside of the cell. So there is a difference in charges from one side to the other. And we actually call this a membrane potential. So a membrane potential is the difference in electrical charges across the plasma membrane. So if you have more of a positive charge on one side and more of a negative charge on the other side, that is going to give you what we call a membrane potential. The resting potential, which is the difference in charges at rest of a cell, usually ranges between minus 10 millivolts and minus 100 millivolts. So to give you some examples from the human body, adipocytes, which store fat, have a resting potential of around minus 40 millivolts, thyroid cells around minus 50 millivolts, skeletal muscle cells around minus 85 millivolts, and cardiac muscle cells around minus 90 millivolts. And I really want you to pay attention to neurons because we're going to come back to this in chapter 12 and spend a lot of time on it. But the resting potential of a neuron is around minus 70 millivolts. The resting potential of a cell represents the potential energy or the potential to do work. So opposite charges attract. So there is an inherent desire for the positive and negative charges to come together. And by keeping them separated like this gives us the potential to do work by allowing the charges to cross the membrane down their electrical gradient. And we'll see in more detail how this works, especially when we get to the nervous system chapter later this semester. Now we're going to briefly talk about the cell's life cycle, which covers the cell cycle and cell division. So we all start from fertilization and one cell, as I mentioned before. When the sperm and the egg fertilize, you get a zygote. But as adults, we have approximately 75 trillion cells. So that is a lot of cell division that occurs. And then even as an adult, you still have a lot of cells that are constantly being replaced. So cell division occurs throughout your entire life. Cell division is the term for cellular reproduction, so that's how one cell becomes two cells. And the lifespan of a particular cell can vary from hours to decades. So you have some cells, like some of your blood cells and skin cells, that may not live very long, whereas you have other cells that are with you your entire life. Apoptosis is a term I mentioned before. This is when you have programmed cell death or a cell commits suicide. There are certain genes that get turned on in that cell that tell it it's time for it to commit suicide. The lysosomes are released inside the cell, which then digest everything in the cell, and the cell is now dead. So before a cell divides, DNA has to be copied. So remember, DNA is the master instruction manual for making all of the proteins. The two new cells that's made from a single cell dividing, both of the new cells need a copy of the instructions. Therefore, before you split two cells or split a cell into two cells, you have to copy the DNA. There are two types of cell division. There is mitosis, which is the division of somatic cells or body cells. This is the majority of the cells in your body. And when mitosis occurs, you start with one cell and you end up with two genetically identical daughter cells. Then there is also meiosis, which is when sex cells are formed, so sperm in males and oocytes in females. You start with one cell and you end up with four genetically different sex cells that only have one set of chromosomes each. So the cell cycle is basically a cell's sort of lifespan and how the things that it does in different stages of its life. So the largest part of the cell cycle is called interphase. And interphase is where the cell is going to spend most of its time, and it is composed of the G1 phase, the S phase, and the G2 phase, which we'll talk about more in a minute. And then you have mitosis, which, as we mentioned earlier, is somatic cell division. So the cell will actually divide into two daughter cells. Some cells also go into what's called a G0 phase, which we'll also talk about more in a moment. All right, so let's start with G1. So in G1 of interphase, this is when a cell is hanging out and doing its normal cell functions. So for example, a pancreas cell would be churning out insulin. This is also where the cell is growing. It can be increasing in cell size. It can be making additional organelles. And it's just doing a whole lot of protein synthesis. The protein synthesis is necessary for both the growth and for its normal functions. Then the S phase is when the DNA is copied. Because remember, we have to copy the DNA before we can divide. So S actually stands for synthesis. So this is when the DNA is synthesized and you make a new copy. So during DNA replication in the S phase, to copy the DNA is unwound because remember it's in a double helix. It unwinds and then there's an enzyme called DNA polymerase which comes in and synthesizes or makes a new strand. And this happens on both strands. So one side is being copied at the same time the other side's being copied. However, because DNA polymerase can only work in one direction, there are slightly different mechanisms for each strand to be copied, and we're not going to go into that level of detail, so that's all I'm going to say about that for this course. Earlier, we talked about RNA polymerase and how it makes RNA, so remember that DNA polymerase makes DNA. All right, so after the S phase, DNA has been copied. Now the cell goes into the G2 phase. During the G2 phase, you're making some last-minute protein synthesis and preparations for cell division. So at this point, you've already copied the DNA, so the cell is pretty much committed to dividing. So these are final preparations to make just before you start the mitosis process. Then mitosis is when you are actually separating those two sets of chromosomes, because remember, you copied the DNA. The DNA at this point is that supercoiled, very tightly wound chromosome structure, and those two sets of chromosomes get separated, and then finally you cut the cytoplasm in half so that each of the two daughter cells get an equal amount of cytoplasm, and the process of actually squeezing the cell in half into two sets of cytoplasm is called cytokinesis. So mitosis is spreading apart those two sets of chromosomes, and cytokinesis is actually breaking the cell into two cells. So you might have had this in high school or another intro bio class, the different stages of mitosis. I'm not going to test you on what happens in each different stage. I just want you to know that mitosis is when the cell is actually dividing. So mitosis is actually the separation of the two sets of chromosomes, and the cytokinesis is the physical separation of the two daughter cells. If you ever do have to remember the different stages of mitosis, I always used to use PMAT to help me remember. So prophase, metaphase, anaphase, and telophase. And then I also want to point out that during this process, those centrioles that we talked about earlier, they move to both sides of the cell. Microtubules play a big role in making the spindle fiber. And this is why I mentioned earlier that if a cell does not have centrioles, it cannot divide, because they are very important to this process. All right, and then there was that G0 phase. So some cells that never divide just stay in this phase. It's kind of like a holding phase, and they just perform normal day-to-day cellular functions. They never are going to copy their DNA, and they're never going to make preparations for cell division. So as an example, neurons and skeletal muscle cells stay in the G0 phase. So these are two cell types that do not divide during your lifetime. So take care of your neurons and take care of your muscle cells. So a little bit more about mitosis. Mitotic rate is the rate at which certain cell types divide. As a general rule, longer-lived cells usually never divide, and again, two examples of these are muscle cells and neurons. And shorter-lived cells divide frequently, and two examples of these are your skin cells and your digestive tract cells. So for cells that do have to divide quite frequently, there is a population of what is called adult stem cells in your body to help maintain the population of cells. So for example, think of, again, of like your skin cells or your digestive tract cells. And what happens is your body keeps this cell that's called a stem cell, and since we're adults, we'll call them adult stem cells. And every time it divides, one of the two daughter cells will stay a stem cell that can keep being used, while the other cell becomes the intended functional cell, like a skin cell or a blood cell or a digestive tract cell. So dividing a cell requires a lot of energy in the form of ATP. So it's a very energy intense process. So if you don't have enough energy reserves, then your cell division is going to slow down or stop altogether. So what does this mean? If you have like a case of starvation where someone is not getting enough nutrients, not getting enough calories, this could result in stunted childhood growth, because you have a lot of cell division that takes place while you're a child growing into an adult. You know, the body itself is getting larger, so you can imagine the amount of cell division going on. So if you have poor nutrition, not enough food, you're going to have stunted growth because you don't have the ATP available. You could also end up with slower wound healing because your skin cells have to undergo cell division to replace the ones that were damaged when you have a wound. So again, if you don't have enough energy in your body, your wounds are not going to heal as quickly. You'll have a lower resistance to disease because your immune cells have to undergo a lot of cell division. You'll learn more about this in 2252, but when you are fighting an infection, there are several types of immune cells that undergo rapid cell division, so you need the ATP energy reserves for that cell division. You'll also end up with thinner skin because skin cells do have to rapidly divide to replace themselves because they're always falling off and getting scraped off. So you're always having to replace skin. If you don't have the energy to do that, your skin will get thinner. The same thing with your digestive tract. Those cells undergo a lot of wear and tear and are constantly being lost. And if you don't have the energy reserves to replace them, you're going to have issues with your digestive tract. So growth factors are extracellular signals that promote cell division. So this is a way that, say, for example, the endocrine system can control cell division in various parts of the body. So there's a hormone, for example, called the growth hormone produced by the pituitary, and that is a signal to certain other cell types in the body that it's time for them to divide. And then there are also internal signals inside all of our cells that control cell division and when it happens and when it should be shut down. And the last thing in this section is about telomeres. So telomeres are segments of DNA on the ends of chromosomes, and they kind of provide a little bit of buffer because every time your DNA is copied, the way that DNA polymerase works is it can't quite reach the end of the chromosome. So every time your DNA gets copied, you lose a little bit off the end. So the telomeres act as protective caps, and every time you copy DNA, the telomeres get shorter and shorter. Now there is an enzyme called telomerase, which can lengthen those telomeres and basically put the protective cap back on. But this enzyme is only active during development, so like when we're embryos. And once we become adults, that enzyme is turned off. And every time our DNA replicates as an adult, it gets shorter and shorter and shorter. This has been hypothesized to contribute to the aging process, so that as your cells divide over time, that telomere, which is shown in this image as the little black caps on the end, gets shorter and shorter and shorter until eventually you actually start losing important parts of your DNA, which leads to all sorts of problems related to aging and eventually death. So when they discovered this, several years ago, they were like, oh, maybe we can fight aging by finding a way to lengthen the telomeres. What if we could turn telomerase on in adults and get those telomeres back? But then they found out that that is actually what cancer cells do. And then that leads us to our next section. So the last section of this chapter is on cancer. So cancer is an illness that's characterized by an accumulation of mutations inside of our cells that disrupt those normal control mechanisms over the cell cycle and over the controls on cell division. So think of cancer as being essentially out of control cell growth, and that's why we're including this in the chapter on cells. So oncogenes are genes that have the potential to affect cell growth, cell differentiation, or cell division when they get mutated. Right, so these are genes that control that cell cycle, control when a cell goes into mitosis, etc. So with malignant tumors, these are groups of these abnormal cells, right, that have these mutations, and we call them malignant once they cause organ and tissue function to deteriorate. So the cells stop doing their normal job. So for example, a group of liver cells that have become cancerous are no longer acting like liver cells, and so that portion of the liver is not doing what it's supposed to. As I mentioned a few minutes ago, cancer cells are able to somehow activate telomerase. Telomerase is active in 90% of all cancer cells, and this allows them to have that uncontrolled growth where they can continue to grow, they can continue to divide, and they have nothing holding them back. There's not just one type of cancer. A lot of times you hear in these TV shows and programs, people talk about a cure for cancer, like there's just one type. But it actually depends on the type of cell that's growing out of control. And this theoretically can happen with any cell in your body, right? So it could be a kidney cell, it could be a bone cell, it could be a skin cell. And then it also depends on the specific mutations that happen inside that cell that cause it to become abnormal. So there are between 100 and 200 different types of cancer, depending on the source you look at and the criteria they use to differentiate one type from another. The mutations that can lead to cancer can be caused by chemicals called mutagens. A mutagen is a chemical that can cause the DNA to mutate. A carcinogen is a mutagen that we know leads to cancer, like for example, cigarette smoke. And then unfortunately, just age and getting older. And that is because, as I mentioned earlier, you've got a lot of cells in your body that continue to divide throughout your lifetime. And the more times they divide, the more chance they have of getting a mutation that turns them into a cancer cell. All right, so here's a little illustration from your textbook. So again, remember, a cancer cell is basically one of your used to be normal cells, but it has become abnormal due to mutations, right? So we start with this abnormal cell shown in purple up here. And then over time, that abnormal cell divides and divides and divides, because remember, one of the hallmarks of cancer is that there are no controls over cell division. So then you get this buildup of abnormal cells, and we call that a tumor. So a tumor is a mass that is produced by abnormal cell growth and division. A benign tumor is a tumor that stays in the tissue in which it forms and shows no signs of having the mutations necessary to cause it to leave that tissue. And it doesn't usually interfere with local functions in that area either. A malignant tumor is one that has gained the ability through additional mutations to be able to spread out into surrounding tissues and then move throughout the body. In the case of malignant tumors, we have some other terminology. So a primary tumor is the original site of the first tumor. So that's where the original cell turned bad and started growing. But then that original tumor can start doing what we call invasion, which is spreading out into the surrounding tissue, where it can actually jump into like your bloodstream or your lymphatic system. And then it spreads to other areas of the body and starts growing there. And that's metastasis, is when the cancer gains the ability to spread to distant tissues and organs. And again, this happens after the cancerous cells have invaded the lymphatic system or the blood vessels, and then you get the formation of secondary tumors, which people in the cancer industry call METs, because the cancer has metastasized. So here's a little illustration just showing you again, luckily, it's not just one mutation that leads to cancer. It usually takes an accumulation of several mutations. So you start with a normal cell, you get a one mutation that might make the cell cycle act a little funny. It's still not necessarily a cancer cell, but it's got an issue. You get another mutation. This might further adjust the cell cycle, but it's still an okay cell. It's still not cancerous. But you get a few other mutations, and then you end up with a cancer cell. So the good news is it does take an accumulation of mutations to become a cancer cell. The bad news is as you age, it becomes more likely because you've got more cell divisions, and every time you get a cell division, you get a chance of another mutation. And then people who have cancer in their families, so a genetic history of cancer, you may like be born with this cell right here where you already have two mutations in place. So people with the cancer in their family history oftentimes are starting out with mutations already in place from day one. So it takes fewer mutations for them to get to a cancer cell. And with normal cells, typically, if there is DNA damage, there are enough mutations, the cell can sense this and can say, oh, something's not right, and the cell commits apoptosis. So that's the cellular suicide we talked about earlier. One of the problems with cancer cells is they have no checks and balances like this. Once a cell becomes a cancerous cell, it's going to continue dividing no matter what. I mean, it can even continue to get mutations, and it doesn't matter. It's going to continue dividing and continue growing. That's what makes it a cancer cell. So here's a little chart comparing the normal cells to cancerous cells. So again, our normal cell is going to have a nice shape. Nuclei is going to look normal. Cancer cells can be large with a lot of variable shapes to them. Normal cells, like all of your liver cells, are going to have a particular look and be arranged in a particular way. But with cancer cells, all that goes out the window. Normal cells have a specific way they should look and function. Cancer cells, again, all that goes out the window. And normal cells actually have a normally pretty slow rate of dividing. And when they do divide, it's highly controlled. And with cancer cells, it's just uncontrolled growth everywhere. So that's all for chapter 3. I know it was a long chapter, so don't wait to start studying. Hopefully a good bit of this was just review from previous bio classes you've had, but there is a lot of material, so I'm going to encourage you to start studying right away.